So what I thought I'd talk about was a grant that was recently funded by 4 AT societies jointly. So just to explain, you know, we had the AT Society had a grant call, and we had a good number of good quality grants, but we were only able to fund one. So there's been a recently formed alliance of AT societies, and I kind of tried to see if we could get funding for at least one of the other grants via that kind of alliance. And so 4 societies, AT Society, Action for AT, IFAT, which is the Spanish society, and Brashat, which is the Australian society, have jointly agreed to fund this particular project. I thought this was kind of interesting to talk about because partly it provides a connection to some of the other ataxias and to protein aggregation, and it focuses on some of the downstream steps. So it sort of brings a lot of things together. It also talks about biomarker analysis, so I could explain to you what that is about. It's not directly going to provide a treatment, so I'm sorry about that, but as you will see, I think there could be things that could come out of it, and we'll go through that as we go through the talk. Okay, so the challenge for AT research is that there aren't animal models of AT to recapitulate the human disease. And I'll talk about kind of model systems and what else we can use maybe another time. But I think that's hampered progress a bit, partly because if we want to be looking at drugs, we do need a model system, but also more importantly, it stops you being able to dissect some of the pathways to really look at mechanism, particularly when we're talking about the brain. But anyway, because we don't have a good animal model, Tanya Paul's group have analysed frozen cerebellum samples from 21 deceased AT individuals and 21 age and gender and race match control samples. And she works in the US, and there they have biobank. So some wonderful families have donated the material, and she's used this for her analysis. And what she's trying to ask is, what is the difference in what we call the proteome? So that's the proteins that are made, the proteins that you can identify in AT versus normal individuals. And then she's also looking at the aggregated proteome, which is the protein aggregates, the kind of association between proteins. And she's found differences, and that's what I'll tell you about. So I'll explain the analysis that they've undertaken, because it's pretty amazing technology, and then the significance of what she's found for the brain signaling, and we'll talk about it in connection with other disorders as we go through. And just to remind you, the region in the AT where there is this progressive degeneration is the cerebellum, which is here, and the particularly important region of the cerebellum is the Purkinje cell layer. So now I'm not going to go through this in detail, but I just want to make sure that the words I'm going to talk about you're going to be familiar with. So we've talked about the DNA code, the linear code that has all the information to make all the cells in the body, built of 4 building blocks. From that, you can extract the information for a subcode, which is called the messenger RNA, and that similar 4 building blocks, but just has the information to make a single protein or worker molecules. And the proteins, they're the worker molecules in the cell. They're built of 20 building blocks, amino acids, and because the amino acids interact very readily with one another, they all kind of assemble and form a complex structure which determines what the function of that protein can be. It's estimated that we have about 20,000 different proteins, or we're able to make 20,000 different proteins. So that would mean we must have 20,000 messengers, and the region on the DNA, which has the information to make any one particular protein, called genes. We have at least 20,000 of those. It's a bit more complex because there can be some alternatives to these things, but we needn't go into that. So when I say we can make 20,000 proteins, not any one cell can make all of those. Different cell types in our bodies will make specialist proteins. So we've got a whole load of different cell types, and of course what we're focusing on is the neuronal cells, or the well, called here the nervous cells, but I think it means the neuronal cells. So what Tanya asked is, are there different proteins present in the AT cerebellum compared to the controls? And in analysing this, what we have to think about is a number of things. I've told you previously that ATM can tag, just drop on a little chemical group onto proteins, and that can change various things about them. But here, it can change their stability, how stable they are, can make them more stable or less stable. And that would, of course, mean that you may or may not detect that protein in these samples. And that may or may not be related to the progressive ataxia. I've talked to you about how if we accumulate DNA damage on the DNA, but that could affect our ability to make the messenger RNA. But that sort of damage, I think, if it was really affecting our ability to make messenger RNA and then protein, should be random. So you shouldn't expect to see defined changes. We're also looking at deceased patients. So the neurons may not be functioning, which could mean that any changes we see are a consequence of ataxia and not the cause of it. So in analysing the results, we've got to think about what are the causes and what might be the consequence. But we'll come back to that later. So to do this proteome assessment, this is really amazing technology, and it's state-of-the-art technology, what they do. And basically, you first want to take the proteins or protein fragments of the proteins from these samples. They're frozen samples. I've told you proteins are very complex, interwoven structures, and there's no way you could really get those structures out of those proteins in an intact state out of frozen cells. But that doesn't matter for what she's doing. What you want to do is have little fragments of them. So you remember I told you proteins are assembled from amino acids, and they're assembled like beads on a chain, really, to give you a peptide, and then they all fold to make the protein. This peptide can be much longer here, and so all you want for this analysis is this string of peptides from a protein. And in fact, you can cut the protein up to give you a number of these peptides. And then you put these little peptides through a machine called a mass spec, which will then tell you the order of these 21 building blocks, these 20 building blocks, the amino acids in that little piece. And because there are 20 amino acids, and it's not going to be such a, not so many proteins will have an identical sequence, you can, by looking at databases, kind of allocate and say you think each one is representative of a certain protein. So just to kind of show you that more graphically, you take the tissue, so she's taken these frozen tissue samples, obtain the proteins, though they're not intact, cut them up, and you've got little peptides, so the little fragments of the protein, but moderately sized fragments, put them through this mass spec machine, and then get the sequence of all these little peptides here. And then going back, and when I say the sequence, I mean the sequence of those 20 building blocks. It's not the DNA sequence like you have on a DNA, but it's reflective of the DNA sequence, because that DNA sequence has the information to tell you which amino acids to put in. And then you go to databases, and you can, from all your little fragment peptides, you can say, oh, that's likely to be such and such a protein. And obviously, often you will have more than one fragment from one protein. So you can work back and say, these are the proteins that are made in a normal brain, and these are the ones being made in the AT brain, or cerebellum, I should say. And she found 3 proteins that are required for something called isophosphate regulated calcium signaling, which are reduced in AT, and that's these 3 here. Now, you may remember I told you neurons, and particularly Purkinje cells, receive and transmit signals regularly. And calcium signaling and the duration of calcium currents are really important for that signaling. I think you kind of get a calcium potential across the membrane. And it turns out that these IP3, so this one here, it's called IP3 receptor, the R stands for receptor 1. The IP3 receptors on the membrane regulate the amount of calcium concentration. This is what we call calcium homeostasis. It's how much calcium is there. And these are highly abundant in Purkinje cells. Then it also turns out that loss of IP3, but I think it might be all of these, but at least some of the genes involved in this signaling pathway cause spinocerebellar ataxia, which are a group of disorders, I think there's 40 of them, which tend to be dominant as opposed to autosomal recessive, which is AT. That means you only need to have one. You remember, you've got 2 copies of the gene. You only need 1 copy inactivated and you have the disorder. But they're quite similar ataxias to AT. So it's significant that these things are missing, and we know that mutations in those can cause ataxia. And from a whole load of studies, it seems like once calcium signaling genes are disturbed, you get abnormal development of the Purkinje cells. And when you go back through the literature, there's other studies that have shown some abnormalities in duration of calcium currents in AT neuronal cells, deficiencies in this sort of metabolism, but it's never really been put together. And, you know, it's the nature of science that people report things, and often that will take off and somebody else will report it, but sometimes it sits there and doesn't do very much. So there's provocative evidence out there, and I think all of this is very provocative, but nobody's really put all this together previously. And this is just to show you kind of what her results look like. So here, this is the amount, somehow they quantify it, the amount of this IP3R in a control, a normal individual, and in the AT cerebellum. And you can see this is the range that's observed. And then these are what they call the error bars, where you work out how significant the difference is. And you can see there's quite a substantial difference. And normally these error bars tell you the error that's in the process. So it could be right down there, could be within the normal range, and up there could be within the AT range. So they're almost non-overlapping, and that's what we call full star significance, quite significant. This one is a little bit less, and you can see these error bars are overlapping, but CA8 is again highly significant. So I think these differences are really quite pronounced and quite significant. So then they also looked at the levels of protein aggregates in AT. And you may be aware that you get protein aggregates are implicated in other neurodegenerative disorders, including Alzheimer's, Parkinson's, ALS, and in some of these spinocerebellar ataxias. It doesn't seem like in these, from what I've read, it's always there. It's kind of implicated, but it's not absolutely there's not always these protein aggregates. So, what she found was there was increased levels of these protein aggregates in AT. And again, that was very definitely significant. So the working model that she's got, and it kind of encompasses the aggregates, is that the loss of ATM causes a toxic protein aggregation, but at the sites of the DNA damage. And so Tanya Paul's worked in the DNA damage response for a long time, and she still thinks there's a connection with the DNA damage. That's particularly important for aprotaxin, AOA1, because this would still relate to DNA damage. So at the sites where there's DNA damage and there may be some abnormal things going on, and she actually has a very nice model for what might be happening, but I'm not going to go through that. You get this protein aggregation, and that protein aggregation may contribute or cause the loss of, particularly the loss of the calcium signaling pathway, and that alone is sufficient to cause the progressive ataxia. Now, obviously, this is a working model. She's seen this. We know there's abnormalities in the DNA damage response, but putting all this together is the challenge, and having a working model is the first step to deal with that challenge. So what she's going to do and what she would like to do is, firstly, her specific model is that it's the neuronal activation that is causing the loss of these calcium signaling proteins. And the reason she says that is because she's got some preliminary data suggestive of that. So basically, as I said, with using this neuronal material, you can't actually dissect and ask specific questions. So she has used a neuronal cell line, and quite a well used neuronal cell line. And firstly, she's able to have ATM being made or not made. So she can ask the difference whether you've got ATM or not. And then they can also activate the neuronal cells, and just by giving something called glutamate exposure. This is quite a well-known technique. A lot of people are using that as a kind of monitor of neuronal activation. And what she's found is that if you add glutamate, activate the neurons, and also remove ATM, you get lethality. And what that is strongly suggesting is that the neuronal activation is a component of the sensitivity of the neurons to ATM loss. And we call this synthetic lethality because if you don't have ATM alone, that's not a problem. And if you activate your neurons, that's not a problem. But doing these two things together seems to be causing the problem. So what she wants to do then is look to see if this neuronal activation and ATM loss impacts upon the calcium signaling proteins, and particularly the IP3R, but the other ones as well. And to exactly try to dissect what that relationship is, how it is that you lose this protein in these samples. The other thing she wants to do is to further exploit those frozen samples. So remember I told you go from DNA to the messenger RNA to protein. And what she's found at the moment is that the protein of these three proteins are reduced in AT brains. So now she wants to ask, are you also seeing a reduction in that code for them? So what you might think is if you don't make the message, and this is what normally happens when proteins aren't expressed, you don't make the message, you therefore don't make the protein. However, if the protein is disappearing or being lost by aggregation or some other type of mechanism, it might be that you're making the protein normally, but something abnormal is happening to it. So she's going to go back and look at what the messages that are made. And that too, I'm not going to go through that technology, but that too is quite difficult technology, although easier than what she did with the proteins. And she's going to use the neocortex as a control because that's not affected in the AT samples. So these 3 specific proteins are all decreased in the AT cerebellum. I think that's not really consistent with a type of random model where you've got protein production. So some of the models I've told you about up to now have suggested that you're not able to sustain making proteins in the AT brain, or you can't sustain making the transcript, messenger RNA, because either you haven't got enough energy because there's energy depletion, or because there's too much DNA damage. I think that is not what is being suggested by these studies. So I think it's important she looks at the messenger RNA to gain that further, and then we need to try and understand really why we're specifically losing these proteins. So the type of model, so the other question is whether this is a cause or a consequence of the ataxia. You know, is it that you get the ataxia and therefore you break down these proteins? I think the fact that mutations in these proteins cause some of these scars, disorders, means at least it can be causal in that situation. Doesn't absolutely mean it has to be in the situation of AT, but I think it's quite likely that it will be given the relationship to these other things. So just to summarize then, the type of model is that you've got these various other disorders that all can result in some form of protein aggregates or abnormal protein misfolding or whatever. And her model for AT is that there's a build-up of lesions or damage responses or something that also results in these abnormal protein aggregates. And I should say that actually there's a strong connection here because all of these disorders also do have elevated damage and have what we call oxidative stress, so these increased reactive oxygen species. So it's possible that there is a connection here, that they're all in some way gaining more lesions in their DNA. That then causes this protein misfolding, protein aggregation. That results in the loss of the calcium signaling proteins that leads to the neurodegeneration. This is only a working model. There are lots of questions. It could, there's possible that loss of the calcium signaling proteins causes the aggregation, but it's not so easy to think of a model for that, whereas this way forward is easier to think about a model. So I think although this is not going to lead to rapid treatment, I think and in some ways it's a research grant to try and understand the link, what's causing ataxia. And I liked it because it's looking in a way at these downstream steps. So she still thinks, and I still think, there's something connected with DNA damage. There's so much correlation with the other ataxias, AOA1, AOA2, something up there, but that somehow is causing these protein aggregates and causing a vital step that we know causes progressive neurodegeneration or causes ataxia. So although it's not going to lead, I think, to immediate treatment, it still could be very beneficial because there are drugs that affect calcium channels and enhance calcium influx through these channels. And I think there's safety in numbers in that if you can sort of show connections of how these other things are all fitting into a common endpoint, it enhances kind of strength in trying to get the drug companies interested in trying to do something about it. But the other point that I think could be very useful here is that it's a bit like the dexamethasone treatment for coronavirus and potentially even for AT individuals. We may not, what we know is that inflammation after coronavirus treatment is causing a lot of deaths, particularly for people that are badly infected in hospital. We don't know how you go from that first step to that second step, but we can still do something about the second step. So even though we may not know how it is you get to this step down here, there could still be ways in which we could try to treat it, to have drugs that will alleviate this downstream step and help, and hence help with the progressive ataxia. And I think that was why all of us felt that this was quite an interesting and a somewhat different grant. Okay, so we'll stop there.